Welcome to this presentation on drug metabolism and excretion. We will explore the processes through which drugs are metabolized and eliminated from the body. Understanding these mechanisms is key for pharmacology and effective drug therapy in veterinary medicine. Let's start with the two primary mechanisms of drug elimination: biotransformation and excretion. Biotransformation refers to the chemical changes a drug undergoes in the body, primarily in the liver, to make it easier to eliminate. Essentially, the body modifies the drug to increase its water solubility, aiding in its excretion. Excretion is the actual removal of the drug from the body, which occurs mainly through urine and the bile. A drug's physico-chemical properties like lipid solubility and degree of ionization play a significant role in determining its fate in the body. These properties influence how the drug is absorbed, distributed, metabolized, and excreted. For instance, lipid-soluble drugs are more likely to be absorbed into tissues and may require transformation in the liver to become water-soluble before they can be excreted. Ionization affects a drug's ability to cross cell membranes. Non-ionized or uncharged drugs can easily pass through cell membranes, affecting their absorption and distribution. In summary, the body must often change drugs from lipid-soluble forms to water-soluble forms to eliminate them efficiently. Sites of metabolism and ester linkages. Metabolism occurs in several parts of the body, each playing a unique role. The kidneys, they primarily excrete polar drugs and drug metabolites. The kidneys filter blood and remove waste products, including drugs. The blood plasma, some metabolic reactions occur directly in the bloodstream. And the lumen of the gut and intestinal mucosa, these areas contribute to the breakdown of drugs, especially those taken orally. And lastly, through the lungs, they are involved in the metabolism of certain drugs, particularly through hydrolytic and reductive reactions. Another important concept in drug metabolism is ester linkage. This is a chemical bond found in certain drugs that makes them susceptible to hydrolysis, a process where water breaks the bond. Drugs with ester linkages include acetylcholine, succinylcholine, atropine, and procaine. These drugs are hydrolyzed by enzymes in the body, which can affect how long the drug stays active and how quickly it's eliminated. Overview of biotransformation. Drugs undergo metabolic changes to transform into metabolites with properties favorable for excretion. The primary goal of these changes is to make the drugs less lipid-soluble and more polar, so they are easier to eliminate from the body. These include the products of biotransformation. After metabolism, drugs typically become less lipid-soluble and more polar, enhancing their excretion. And hydrophilic drugs are usually excreted without significant metabolism. For example, streptomycin and stigmine are excreted largely unchanged because they are already water-soluble. Drug metabolism is divided into two types of reactions: phase 1 reaction and phase 2 reactions. These phases are crucial in converting drugs into forms that can be easily excreted. Phase 1 reactions include oxidative, reductive, and hydrolytic processes that modify drug molecules. These reactions introduce or expose polar groups on the drug molecule, such as OH, SH, COH, and NH2. In order: hydroxyl, sulfhydryl, carboxyl, and amino groups. By adding or exposing these groups, the drug becomes more water-soluble and ready for further modification or direct excretion. For example, oxidation adds an oxygen atom to the drug, often making it more reactive and polar. Reduction adds hydrogen or removes oxygen, altering the drug's structure. Hydrolysis breaks the drug into smaller, more water-soluble pieces. These modifications are essential for preparing the drug for phase 2 reactions or excretion. And finally, phase 2 reactions are also known as conjugation reactions, involve attaching endogenous substances to the drug or its phase 1 metabolites. These endogenous substances include glucuronic acid, acetate, sulfate, and amino acids. The purpose of these reactions is to make the drug more water-soluble and typically inactive, ensuring that it can be easily excreted from the body. In simple terms, phase 2 reactions make the drug stickier for water so it dissolves better in urine or bile, allowing for easier elimination. It's important to note that phase 1 metabolites can be either active or inactive, meaning they can still have some therapeutic effects or be completely inert. Phase 2 metabolites are generally inactive and solely prepared for excretion. These two phases work together to ensure that drugs are processed and removed from the body efficiently, minimizing the risk of toxicity and ensuring that the therapeutic effects are controlled.
At this point, let us dive into various oxidative reactions of drugs, which are a major part of phase 1 biotransformation reactions. These reactions are classified as non-synthetic, meaning the drug is chemically modified without adding any new molecules. The result can be an active or inactive metabolite, which affects how the drug works in the body. Oxidation is one of the most critical reactions in drug metabolism. It involves either adding oxygen or a negatively charged radical or removing hydrogen or a positively charged radical from the drug molecule. This process is primarily carried out by microsomal oxidases in the liver. The main player in this system is a group of enzymes called cytochrome P450. This enzyme works with other components like NADPH and oxygen to modify the drug. These reactions help transform the drug into a form that can be more easily excreted by the body. Here, we see an illustration of the hepatic microsomal drug oxidizing system, which is vital for drug metabolism in the liver. This system requires three main components to function. Number 1, NADPH. It provides the necessary energy for the reaction. Number 2, oxygen. This is essential for the oxidation process. And number 3, the substrate or the drug. This is a compound that undergoes oxidation. The core of this system is the cytochrome P450 enzyme, which can switch between two forms, which are A, the oxidized form. This is the inactive state of the enzyme. And 2, AH2 or the reduced form. This is the active state, which facilitates the addition of oxygen to the drug. Here, you're able to identify the various oxidative reactions, which include the oxidative reaction, the drug, the metabolite. And we have to know that these are pharmacologically active compounds. While oxidation is a major pathway, reduction reactions also play a role in drug metabolism, although they occur less frequently. Reduction involves the addition of hydrogen or the removal of oxygen. This process can happen in drugs containing groups like disulfide, azo, or dinitro. These reactions are the reverse of oxidations but also include cytochrome P450 enzymes working in the opposite direction with the help of reductases. Even though they are not as common as oxidation reactions, reductions are essential for certain drugs' metabolism and elimination. Examples of reductive reactions include the inactivation of chloramphenicol by ruminal microbes. This is why the oral route is not suitable for administering this drug to ruminants, as their gut bacteria can inactivate it before it can be observed. Understanding these reductive reactions is important for predicting how different drugs will behave in various animal species, as not all animals have the same metabolic pathways. Hydrolysis is also another significant metabolic pathway, particularly for drugs with an ester linkage or an amide bond. During hydrolysis, the drug molecule is split into smaller fragments by adding water, which makes it easier for the body to eliminate. This reaction can occur in several body parts, including the liver, intestines, plasma, and other tissues. Hydrolysis is crucial for breaking down complex molecules into simpler, more water-soluble forms. Here are some examples of drugs that undergo hydrolysis. Number 1, acetylcholine. These are broken down by the enzyme acetylcholinesterase. Number 2, succinylmethonium, metabolized in the plasma by pseudocholinesterase. Atropine, these are hydrolyzed in the plasma by atropinase, which is why rabbits can safely consume the plant Atropa belladonna. Other examples include procaine, which are metabolized by plasma cholinesterase, and lignocaine, which are broken down by non-microsomal hepatic amidase.